Reporter gene. While the squirrel marker, also known as the reporter gene, can be detected visually directly or following a biochemical assay, in terms of color or individual signal in the transgenic cell or plant. They may be attached to the gene of interest or to regulatory sequences and indicate at which stage and where transgene expresses in the plant tissue both transiently when standardizing transformation protocols for a genotype or in cases of stable transformants. Thus, they perform three functions: reconfirm the success rate or transformation status of the tissue grown on selection media during both transient and stable transgene expression, transform or not localize the transgene expression as to which is the exact region of the protein expression, and important means of screening in situations with high transformation frequencies. A good reporter gene should be stable, sensitive, and not specific to any tissue or genotype, i.e., reliable to work within a range of cells and species. E.g., of some reporter genes commonly used during plant transformations, each having its own positive and negative traits are beta-glucuronidase GUS, figure 12.46 green fluorescent protein GFP, chloramphenicol acetyltransferase, CAP, luciferase, LUC, and table 12.11 and table 12.12, and based on the nature of study at hand, a specific reporter gene is chosen. GUS and GFP are the most commonly used. Dual reporter systems are also made, e.g., GUS, Luke. In the last 3 decades, one of the most beneficial researches in the field of biological sciences has been the isolation of DNA that makes jellyfish, Equirria victoria, glow fluorescently, for which Chalfie and Chen received the Nobel Prize. Bioluminescence in this crystal jellyfish is due to two proteins, acorn, a photoprotein, and green fluorescent protein GFP. The discovery of GFP has brought forth a revolution in cell and molecular biology and transgenic research. Figure 12.46 GUS, reporter gene, assay performed after transformation. A GUS assay was performed on the groin-filtrated tobacco leaf tissue, after being put on a selection and regeneration medium. Arrows indicate the putatively transformed cells stained blue against the non-transformed cells that appear green. B. After floral dip experiments, the inflorescence arrow, Arabidopsis thaliana, appeared blue following GUS assay. Some of the cells of Siliqui also gave a positive coloration. Genes, enzymes, external. Substrates, role of gene. Latch beta-galactosidase ONPG, X-gal bacterial transformation, transport and metabolism of lactose in E. coli. We get GUS A. Beta-glucuronidase MUG, X-LUG most exploited reporter gene, screens gene delivery system, classical. Promoter activity assay, and protein. DNA interaction studies. Luciferase luciferin bioluminescence, assesses the transcriptional activity in cells. LUXA, B-LUXF. Decanal codes for protein in bioluminescent bacteria. GFP green fluorescent protein, GFP. Non-revolutionary discovery, codes for protein in jellyfish, non-destructive reporter gene, and also used as biosensors. R, C1, B anthocyanin and pathway regulatory factors. Non-anthocyanin biosynthesis in vegetative tissues. Table 12.11 non-selectable marker genes or reporter genes. Reporter gene. Response in. Transformed tissue and property. Advantage disadvantage. GFP glows green when exposed to ultraviolet light. Several spectral. Variants are now available for assessing. Multi-transgene. Expression by mutation. A GFP gene. Composed of 238 amino acids out of which some impart chromophore character. Like serine, tyrosine N. Glycine. No destruction of tissue and can measure gene expression. Manual separation of transformed and non-transformed cells, time-consuming and tedious, requires a fluorescence microscope. Reporter gene. Response in. Transformed tissue and property. Advantage disadvantage. Luciferin solution substrate treatment. To tissue in presence of. ATP causes oxidation of. Luciferin emits light. Non-toxic manual separation of transformed and non-transformed cells, time. Consuming and tedious. Use a range of substrates available depending on. Assay like colorimetric. Fluorometric R. Histochemical type. Cells metabolize X-LUC. Source of carbon N. Energy. After overnight. Assay 24 hours at 37 OC. With substrate X-LUC. 5-bromo-4-chloro-3. Indole beta-D-glucuronic. Acid methanol N. Buffer, transformed. Tissue stains blue. Treatment with 100% alcohol removes. Chlorophyll pigment N. Enhances the clarity of blue spots. Excellent for detecting and localizing even a single cell without any sophisticated equipment. Fluorometric. Assay with 4-methyl beta-D, glucuronide MUG. Destructive assay method causing cell death of tissue. Table 12.12 properties of different reporter genes demonstrated in transgenic plants. Methods for gene transfer. Production of genetically modified GM crops has proved to be a remarkable milestone in the history of crop science. Although always controversial, the GM crops have surely created a niche for themselves. Remarkable success has been achieved in the last 3.5 decades of transgenic research, but it has remained confined to a group of species that render themselves amenable to genetic modification. This fact and requirement to enhance transformation efficiencies underlines the necessity to develop new protocols or improvise the existing techniques of DNA delivery into various plant systems. In order to transfer alien or foreign genes into plant cells for GM crop production, 3 major membrane barriers need to be breached: the plant cell wall, the cell membrane, and the nuclear membrane. This can be assisted by a biological agent or vector, or can be achieved either by physical or chemical treatments. The former should somehow gain entry and get integrated, while the latter should create pores in the cell wall and membrane, large enough for DNA entry while avoiding leakage of cytoplasm sonication-assisted agrobacterium-mediated transformation (SAAT) techniques. The following sections emphasize some major transformation methods used for plant transformation. Vector-mediated. A vector is a carrier that delivers the GOI into a target cell for replication and transgene expression. As discussed earlier, ORI origin of replication MCS, multiplying site, and the selectable marker gene are the 3 components of any vector. The ORI gene enriches the region where replication initiates by unwinding of the vector, making it accessible to the polymerases for replication. The MCS or restriction or recombination site carries some unique sequences that can be recognized by specific restriction endonucleases permitting site-specific exchange between two plasmids, and thereby insertion of gene of interest GOI. The selectable marker gene serves its role in ensuring insertion of the vector into agrobacterium. The 2 plasmid vectors mediating plant transformation are tumor-inducing T plasmids and viral-based vectors. Tumor-inducing T plasmid and root-inducing Greek plasmid. Agrobacterium tumefaciens, a soil-borne plant pathogen, infects a range of Dictyphilodonus species. It is known for causing crown gall disease or a kind of tumor in dicots credited to the tumor-inducing plasmid PTI. It harbors and has been endowed with a very unique gift as well. The same goes for other agrobacterium species, e.g., a rhizogenes, a soil-borne gram-negative bacterium harboring root-inducing plasmid pre, and known to cause hairy root disease in dicots. They are nature's wonder wherein both plasmids have the innate ability to transfer part of their DNA into host genome and carry out genetic engineering or modification. Also, within the tDNA region of both the plasmids, some genes with regulatory sequences are present that can be recognized by plant genome, while other genes have prokaryotic regulatory sequences. They have brought forth a revolution in the scientific world of biotechnology, earning them an apt. And keeping in mind that the damage is not irreparable and membrane can overcome the shock. The DNA to be introduced is manipulated in the laboratories prior to delivery. At last, it should reach the desired destination, i.e., the nucleus for integration into the plant genome and stable inheritance. Naked introduced DNA which is unfolded and without proteins cannot survive for long outside the nucleus. It should get integrated with the natural protein synthesizing machinery of the native DNA in order to be retained and transcribed. Genotype to be altered is a prime factor responsible for the selection of a particular method of gene delivery. A lot of effort goes behind the standardization of genetic transformation technology for crop species, but once established, the procedures can be routinely used. Recovering a transgenic plant is an extremely challenging and perseverant task that can be hindered by the in vitro recalcitrance of plant species, thus requiring optimization at every step. Selection of the appropriate genotype and the apt target tissue that has good regeneration potential, i.e., tissue culture friendly, efficient method of gene delivery and screening methods of putative and stable transformants are all important steps towards recovering a genetically stable transformant. Selection of the appropriate genotype and the apt target tissue that has good regeneration potential, i.e., tissue culture friendly, efficient method of gene delivery and screening methods of putative and stable transformants. Selection of the appropriate genotype and the apt target tissue that has good regeneration potential, i.e., tissue culture friendly, efficient method of gene delivery and screening methods of putative and stable transformants are all important steps towards recovering a genetically stable transformant. The method of gene delivery can be mediated by vector, e.g., T plasmid of agrobacterium or virus mediated, which are indirect and biological means of transformation. In addition, direct delivery systems of particle bombardment, microinjection and electroporation protoplast mediated constitute the physical means of transformation. Amongst available methods discovered for transgenic production, agrobacterium mediated transformation and bioelastic gene gun or microprojectile bombardment or particle bombardment remain the methods of choice and are widely employed depending on the plant species and the target tissue. With the advancement in technologies and in order to enhance transformation efficiencies, modifications of methods have also come up as in the case of germline transformation like MEG-LITR, male germline transformation, floral dip employing agrobacterium, agrolistics, and plasmids. Based on the type of opine produced, the T plasmids are categorized into different types like octopine, novoline, or succinopine. Plasmid having the same opine type may still differ in the gene content. Upon infection, it delivers only tDNA portion that is flanked by 225 base pair repeat sequences forming the left border LB and the right border RB. LB is not essential, but RB is vital for transfer of DNA and initiates the integration of the tDNA region into the host. So, any DNA or GOI within these LB and RB is only transferred when a GOI infiltration is done. The genes in tDNA region are controlled by regulatory sequences like promoter and terminator and have low G plus C content apt for expression in host cell. Transfer of large DNA sequences is made possible by omitting the T vector backbone sequences. The pre-share functional homology with the PTI. Even in case of pre, the Vir genes are essential for transfer of tDNA into plant. Title of Nature's Genetic Engineer. Amongst them also tumefacines, also classified as rhizobium radiobacter, is the most exploited for plant transformations. Agrobacterium-mediated gene transformation, commonly termed a growing filtration, is the most common method of transformation and is controlled by a unique gene transfer mechanism. Size of both PTI and free is nearly 200 kbp. Along with an origin of replication, T plasmid has three main regions. The transfer DNA region tDNA, approximately 24 kbp in size. The virulence region encoding the Vir genes Vir A, B, C, D, E, F, and G grouped into operon 0 after encoding for proteins mediating the excision. Conjugated transfer and integration of the tDNA into the host genome. Opine catabolizing region figure 12.47. The tDNA region also contains gene responsible for the biosynthesis of opine, main carbon source, phytohormonoxin, and cytokinin responsible for tumor or gall formation. Additionally, many unique gene sequences are present in both. Figure 12.47 tumor-inducing T plasmid, approximately 200 kbp in size, of agrobacterium tumefacines. Sunflower tissue using a tumefacines. While first transgenic plants were generated, were tobacco, in the year 1984. Agrobacterium is normally cultured in YEB medium at 28 OC. If cultured above 28 OC, the bacteria lose the T plasmid. Sucrose, surfactant forms important constituents of the culture medium. Compounds like acetylzurindone are added to infiltration medium to mimic natural process and induce virulence in the bacteria. This phenolic compound is naturally produced by the cod plants upon infection, inducing the virulence of the bacterium. Agrobacterium strain, number of applications of infiltration media, humidity chamber during co-cultivation, and target accessibility are all important deciding factors affecting transformation efficiency. Being a comparatively simple and convenient technique without the requirement of any sophisticated equipment, it is a very efficient method already standardized for a variety of plant species and target tissues. It results in single copy insertions or low copy number transfer that is desirable for stable transgene expression. Major limitation is the plant genotype dependency and bacterial host range specificity. Virus mediated. Viruses are acellular, non-living entities that can reproduce only inside the living host cells. After infection, millions of identical copies of the virus are replicated inside the host wherein new viruses are packaged and assembled. Packaging of foreign DNA into a virus is not that amenable compared to a bacterium. They can also be used to mediate gene introduction naturally owing to this pathogenic nature towards plants. Retroviruses also can produce high viral titers and have great stable transduction rates both in vitro and in vivo. Upon being converted to cDNA, the viral genome can easily be manipulated owing to its small size. Their entry into host is very simple and can be achieved by merely rubbing a leaf with the viral inoculant. Due to replication in the transduction machinery, they spread throughout the host even if infection starts from a single cell. Viral gene expresses throughout. The figure gives a detailed depiction of all the genes present in a T plasmid of agrobacterium tumefacines. Along with an origin of replication, T plasmid has three main regions. The transfer DNA region tDNA, approximately 24 kbp in size. The virulence region encoding the Vir genes Vir A, B, C, D, E, F, and G also having the region for excision. Conjugated transfer and integration of the tDNA into the host genome TRA genes and opine catabolizing region. The tDNA region contains the genes involved in the synthesis of auxin, cytokinin, and opine, and is flanked by left and right border each made of 25 base pair repeat sequences. As we have already discussed, the vector is modified for transformation experiments. The tumor-inducing genes are removed known as disarming the vector. Thus, the T plasmid we use for carrying out transformations has a modified tDNA region where the tumor-forming genes are replaced by GOI transgene and marker gene. As a result, the vector becomes large size, which is difficult to handle. This led to the formation of two kinds of vector systems, pointed rather than binary vectors. In co-integrated vectors, homologous recombination occurs between an intermediate vector harboring the GOI E. coli plasmid and disarmed T plasmid without Vir genes, resulting in a large co-integrated vector that can be introduced back into agrobacterium. Still, size remains a challenge in the scientific finding that tDNA can function independently of the T plasmid paved way for the development of binary vector two plasmid system. In binary vector there are two plasmids, a helper plasmid wild type PTI without tDNA region, and a mini plasmid small size plasmid having tDNA, Oreo both E. coli and a tumefacines put together into agrobacterium. Helper plasmid carries all genes required for gene transfer and integration. The two Oreo allow shuttling and cloning in both bacteria. Transformed agrobacterium is used for transformation experiments. Using the growing filtrations, prokaryotic genes were occasionally transferred and made to express. But in 1983, the first report of transfer and expression of a eukaryotic gene encoding seed storage protein fasolin was established in which microprojectiles coated with DNA were fired under vacuum and sheer force of high-pressure helium gas. This is the second most widely employed method and a promising one even for those plants that are refractory to other methods of transformation such as agrobacterium-mediated. The device that carries out this bombardment is known as a gene gun, or particle gun, or bioelastics, or ballistics, or bioblaster. A common procedure for any transformation experiment is that the gene of interest is isolated from a source organism and inserted into a transgenic construct under the control of regulatory sequences like promoter and tagged with a marker. This construct is inserted into a plasmid. Microcarrier or microprojectiles 1-2 micrometer sized gold or tungsten particles are coated with the desired engineered plasmid DNA to be introduced in presence of calcium chloride and spermidine that helps in the binding. The metal chosen is inert that will itself not react once inside the plant tissue. The coated metal particles are then loaded onto a macrocarrier film which ruptures upon the shooting of the gun in vacuum and under high helium pressure, accelerating the microprojectile's metal with DNA towards the suitable target tissue placed underneath. Electrostatic energy of a drop of water when exposed to high voltage also helps in this acceleration. This brute force enables the microprojectile coded DNA to penetrate any physical barrier to carry out transformation and integrate with the chromosomal DNA of plants followed by selection and regeneration of plant tissue. Figure 12.48. The host and that is analogous to transgenic cell production. Transformation efficiency achieved by them can be high, but the introduced foreign gene does not integrate into the host genome and thus does not get inherited. Viral vectors are thus profitable, especially in cases where overexpression of a particular protein is the aim and not the complete transgenic plant production. Direct DNA transfer. Physical method agrobacterium-mediated transformation is the most popular method of transgenic production, but is the preferred host of agrobacterium as dicotyledonous plants. They have not been as successful for monocotyledonous plants. This paved way for alternate methods of direct gene delivery into plant cells without the use of any biological agent. Direct DNA transfer can be by different ways. Each has benefits and drawbacks like plain DNA inhibition by cell, physical means or chemical means. Physical methods consist of particle bombardment, electroporation, microinjection, sonication, pollen tube pathway, liposome-mediated transformation, liquefaction, silicon carbide fiber-mediated transformation, and peptide nanocarrier-mediated transduction. The most common direct delivery method of these is bialistic gene gun or microprojectile bombardment or particle bombardment. Bialistic gene gun or microprojectile or particle bombardment. A genotype-independent tool of gene delivery was developed by Sanford et al. 1987 in. Figure 12.48 direct delivery system by particle or bialistic bombardment. A. Bialistic registered PDS 1000 P system of Biorad, USA. Tabletop or laminar hood model picture courtesy. Website. B. Helios registered gene gun system of Biorad, USA. Handheld gene gun model picture courtesy. Biorad website. C. Working of bialistic registered PDS 1000 P system. Microcarrier or microprojectiles 1-2 micrometer size gold or tungsten particles are coated with the desired engineered plasmid DNA to be introduced. The coated metal particles are then loaded onto a macrocarrier film which ruptures upon shooting of the gun in vacuum and under high helium pressure, accelerating the microprojectile's metal with DNA towards the suitable target tissue placed underneath. The main advantage of this technique is that gene gun is independent of genotype and can be used for any kind of plant tissue, be it leaf, meristem, shoot and root, pollen grains, and immature or mature embryos. It has served to be a boon for the transformation of monocots, especially cereals. The major disadvantage is the excessive injury of target tissue owing to high bombardment velocity and insertion of multiple copies of the transgene at the same site that sometimes may lead to transgene silencing in the progeny. Also, in the earlier model of the gene gun, figure 12.48a, excision of the target tissue and a good regeneration protocol was a prerequisite. But with the development of the handheld gene gun model, figure 12.48b, that problem is solved to some extent. Of the electric field and exposure time of pulse according to cell size, electrocomposition in the electrocuvette, pulsing medium, addition of adjuvants like polyethylene glycol, pre and post-pulse manipulation like prior heat shock treatment given to cell protoplast or cold temperature treatment given after pulse shock. The plant genotype is also important in terms of the recovery part or post-pulse regeneration. Unlike agrobacterium-mediated transformation, host specificity is not a limitation for transformation by this technique. Microinjection. It is a direct physical DNA delivery technique to inject the desired exogenous DNA using a fine injection needle directly into the protoplasm or the nucleus of a plant cell followed by regeneration of the transformed tissue. Figure 12.49. It is an excellent alternative for transforming single cell systems like pollen grains or egg or embryo cells, ensuring enhanced chances of transformation, but is quite labor intensive and consumes a lot of time and skill to handle single cells. Also, this becomes a limitation that this method can yield few transformation events at a time. The post-injection handling and culturing of the cell again requires optimization. Electroporation. Another popular and effective physical method of direct gene delivery that introduces desired gene into protoplasts is electroporation. As the word literally suggests, it refers to the creation of temporary pores in the membrane through electric shock facilitating the entry of any foreign material like DNA. For this, the cell protoplasts once isolated are transferred into an electrocuvette in presence of an ionic solution and the desired DNA vector. This is followed by the application of a brief pulse of high voltage to the cuvette. This creates transient pores in the membrane for a fleeting amount of time facilitating the movement of DNA across it. Upon availability of suitable nutrients in the culture medium, the cell membrane heals up and pores reseal. Cells regenerate into transformed callus or plantlets. However, many parameters may affect transformation efficiency and recovery of transformed tissue like the vector use, electroporation conditions, or the plant cell to be transformed. As far as the construct is concerned, the type of construct, linear or circular, its concentration and regulatory elements attached to the gene of interest influence the efficiency. The various electroporation conditions also need to be standardized for each event like pulse strength. Figure 12.49 microinjection. Nuclear membrane transforming a cell. As just mentioned, the second component of the cell membranes, besides lipids, is the protein. So, gene delivery via protein transduction domains or cell-penetrating peptides, CPPs, or peptide nanocarriers has also been reported in which short peptide sequences have been actively transported across the membranes mediated by these CPPs. Till now, the detailed mechanism is not known in depth. Even materials like silicon carbide fibers have been used to mediate transformation, in which they are added to a mixture of the plant tissue and foreign DNA and agitated by vortex mixing. It is known as silicon carbide fiber mediated transformation, SCMT. The collisions between tissue and fiber results into small holes in the target tissue through which foreign DNA-coded fibers can gain entry. Although it is less cumbersome and inexpensive, providing an alternative to the normal methods of transformation that transforms with less efficiency, sometimes causing irreparable damage to the cells. At the same time, silicon carbide has carcinogen. At the same time, silicon carbide has carcinogenic properties that do not make this method popular. With their limitations, all these discoveries have surely been great innovations that provide simple, cost-effective alternatives to genetic manipulation, especially in those plant genotypes that are not easily accessible to modification by the other means. Direct DNA transfer. Chemical method of direct DNA delivery through chemical means works for many genotypes and is based on the fact that when protoplast is incubated with DNA in presence of a buffer having membrane destabilizing or precipitating agents, foreign DNA can gain entry causing transformation. Some popular agents are polyethylene glycol, PEG divalent cations like calcium, poly-L-ornithine, and polyvinyl alcohol. Polybrane spermidine or polycation DMSO technique. Polycation-mediated transfection of cells is quite common for bacteria and mammalian cells. In literal terms, they are cations with more than one positive charge. A direct physical DNA delivery technique to inject the desired exogenous DNA using a fine injection needle directly into the protoplast or the nucleus of plant cell followed by regeneration of the transformed tissue. Sonication. The sound waves can also be applied to create agitation in membranes of tissues and facilitate the movement of particles or exogenous aid through them. This is due to a process known as cavitation that is created by the high-energy ultrasound waves. Although it requires a specific instrument for the purpose, in combination with other methods like microinfiltration, known as sonication-assisted agrobacterium-mediated transformation (SAAT), it has been employed to enhance transformation efficiency. Other methods. There are many more ideas that have been successful in converting theoretical concepts into results. One of them is germline transformation through the pollen tube pathway. The exogenous DNA can be applied to cut end of style after pollination and reach the ovule along with pollen tubes. The exogenous DNA can be applied to cut end of style after pollination and reach the ovule along with pollen tubes via the pollen tube pathway, or the transformed pollen can be applied to stigma for successful fertilization and sire of seed having the introduced soy in the genome. Either way results in the seed with the transgene and has great applicability in modern agriculture. Another efficient method of direct gene delivery, although not routinely used, is lipoaction transformation mediated by the liposomes, first described in 1980 by Fengler. Liposomes are spherical lipid entities formed by the hydration of phospholipids with an aqueous environment inside where they can carry water-soluble molecules like DNA. Cell membranes are made up of phospholipids bilayers with proteins and thus are lipophilic in nature. Any lipid molecule can fuse with the membrane. So, the liposome with foreign DNA adheres to and fuses with the membranes transferring the exogenous genetic material in this process into the cell. In the same manner, if in process the whole effort may become futile. It is rather a significant step in any plant transformation technique. If in any method transformation frequency obtained is higher than the screen, not much of a problem. But in cases where the frequency obtained is low, one cannot afford to waste tissue in destructive analysis. Also, it is not cost or time effective to screen every shoot that regenerates individually. So, it becomes all the more important to have a correct selection system. That is why a selectable marker or reporter gene is inserted into the transformation vector. In the former case, a selective agent is added to the culture medium that supports the growth of only the transformed tissue. A good selecting agent should work at very low concentrations that vary from genotype to genotype due to different sensitivities. It is rather preferable that it inhibits the growth of non-transformed cell and not kills it. While in the latter case, visual proof of a successful transformation event can be seen both by destructive or non-destructive analysis. An ideal reporter gene should be non-reactive to plant tissue and have an easy, highly sensitive, effective detecting system with a preferably non-destructive mode of assay. Thereafter, many biochemical analyses exist to screen transformed tissue for successful transgene integration like tissue PCR, southern blotting, northern blotting, and immunological methods like ELISA analysis. Transformation efficiency is also affected by the silencing of genes, unpredictable transgene expression and growth of chimeric plants. With the advancement of research and awareness amongst people, there also arises the need to selectively remove marker genes to generate marker-free plants. In the following section, these topics will be discussed. Gene silencing. One of the major concerns while creating transgenic is the segregation pattern and stability of the transgene. Many times, in spite of having an extremely efficient transformation experiment, the result may not be as expected. The segregation pattern deviates from the Mendelian ratios. Copy number of the introduced. For macromolecules with multiple cationic sites and can be employed as nucleic acid or protein delivery agents. In aqueous medium, they form complexes with DNA because of the electrostatic binding of oppositely charged double helix. The polycation can covalently attach to a receptor recognizing signal that selectively binds to a receptor and allows uptake of DNA, e.g., polylysine. Even in absence of binding with a receptor, some polycations allow for transfection of cells, e.g., poly-N-ethyl-4-vinylpyridinium bromide (PDBP). Polygreen, a less toxic polycation, increases the absorption of DNA to the surface and is many times used in combination with other chemicals like spermidine or dimethyl sulfoxide (DMSO). Spermidine is a DNA protectant and does not allow DNA to shear because of its condensation effect. DMSO enhances foreign DNA uptake by increasing membrane permeability. Calcium phosphate precipitation. In isotonic phosphate buffer, the divalent calcium ions from calcium chloride solution precipitate DNA, which is exposed to cells to be transformed in combination with physiological shock. PEG-mediated gene transfer. Treatment of polyethylene glycol at molecular weight 4000 PEG-4000 with V in combination with osmotic like mannitol and calcium nitrate solution when given to protoplasts in presence of exogenous DNA enables uptake of foreign DNA into the cell. DDAE dextran procedure. High molecular weight diethyl amino ethyl DDAE dextran was the first transfection reagent to be made. It has also been used to carry out transformation as it is a quite affordable and simple procedure, although it does not produce stable transformants. Being polycationic, carbohydrate interacts with DNA by electrostatic interactions and the entire complex carries a positive charge. This further interacts with a negatively charged cell membrane and gains entry into the cell by endocytosis. Monitoring expression of transfer gene as much as improvising a good transformation protocol is important. It is equally imperative to devise an efficient screening method for monitoring the transgene expression, otherwise known as cis inactivation. While it is the result of the effect of one locus on another, it is transtype, i.e., a transgene can get silenced if present with a silenced homologous copy. Some situations that can result into low expression or gene inactivation. Regulatory elements near the site of insertion can influence expression. Silencing associated DNA methylation. Chromosomal environment of transgene and repeat-induced methylation. If insertion occurs near heterochromatin or repetitive DNA sequences, silencing can result. Copy number of transgenes per integration site also influences expression. Multiple copies exist. They can be present integrated as direct repeat head to tail or inverted repeat head to head or tail to tail. Latter, i.e., inverted repeats show low expression levels indicative of gene silencing. This is the reason why transformation events with single copy inserts are preferred. DNA-DNA interactions and gene silencing in trans. One silenced gene locus can silence another homologous locus. Gene silencing or knockdown occurs by some kind of obstruction during or after transcription called transcriptional TTS or post-transcriptional PTGS respectively. The modification can be in DNA or chromatin structure that affects transcription or even results in change in protein conformation. DNA modification brought about by DNA methylation is part of transcriptional gene silencing. The copy number of transgenes, presence of repeat sequences in the transgene inserts, and production of unusual RNAs can also bring gene silencing. In post-transcriptional cases, RNA involvement has been observed called RNA interference or RNAi mechanism, figure 12.50. Gene may not correlate with equal expression levels. The unexpected behavior of transgene and putative transformants after any event is not only due to copy number variation or the effect of regulatory elements. All this is indicative of some endogenous mechanisms that may be under epigenetic control and influence expression levels. Epigenetics refers to above genetics. In other words, it is a heritable changes in gene function which is not elucidated by alterations in the DNA sequence but rather a consequence of modification of DNA, RNA, or histone proteins. Normally also one can witness the phenomenon of epigenetic gene silencing in plants which is mostly post-transcriptional. The same wonder of gene inactivation or silencing is found in genetically modified plants. It is a natural defense response of host genome against any transposons, be it a transgene or virus attack. In transgenic plants too, the introduced genes can get silenced after integrating into the host genome. It is mainly due to the presence of a transgene homolog known as co-suppression when both transgene and endogenous gene get inactivated. It is more enhanced in haploids and homozygous plants in comparison to hemizygous plants suggestive of a transgene dosage effect. E.g., a co-suppression of genes involved in tomato fruit ripening and flower pigmentation as in the case of petunia. Chalcone synthase CHS gene gets silenced. Polysaccharides are the carbohydrates that upon hydrolysis yield multiple monosaccharides. These are long-chain molecules formed by glycosidic linkages formed between several monosaccharides. When composed of a release coating. Polysaccharides are the carbohydrates that upon hydrolysis yield Regardless of the method, be it direct or indirect DNA delivery, the number of copies of the foreign gene that get integrated and the position of insertion cannot be defined. Trans S can get integrated at any site S in the target genome. If silencing results from integration of foreign gene S at a locus situated in or near hypermethylated sequences, it is Figure 12.50 RNA interference RNAi mechanism or antisense RNA technology, a post-transcriptional gene silencing PTGS system. The flowchart gives an account of the process involved in RNAi mechanism. It is a conserved biological response to dsRNA, governing resistance to both endogenous and exogenous pathogen attacks, thus regulating gene expression. The microRNA, mRNA, and small interfering RNA, siRNA, are central to the RNAi mechanism. RNA silencing is a broad range of host defense mechanism that causes the degradation of homologous RNA. It has been observed in plant, animal, and fungal kingdoms. A prime feature is the presence of microRNAs, mRNAs, and small interfering RNAs, siRNAs, on which enzyme dicer RNase 3-like enzyme can operate. Major differences between TGS and PTGS are tabulated below. Table 12.13. Property transcriptional gene silencing TGS, post-transcriptional gene silencing PTGS. Results from failure of initiation of transcription through methylation and/or chromatin condensation. Breakdown of cytosolic mRNA. When abnormal sense, antisense, or double-stranded forms of RNA are produced, mRNA fails to accumulate. Promoter inactive by excessive DNA methylation and/or heterochromatization. Active. Association with DNA methylation often sometimes. Property transcriptional gene silencing TGS, post-transcriptional gene silencing PTGS. Heritability may heritable get reset. Affected genes get reactivated after meiosis. But in affected lines, it recurs once in every generation at some stage. RNA involvement not the mRNA and siRNA involved. Table 12.132 types of gene silencing are found, TGS and PTGS. Off late, science has seen a lot of progress in the molecular understanding of epigenetics and epigenetic role in gene silencing. Discovery and involvement of siRNA, mRNA have opened avenues that were never trodden on before. Transgenic research provides a good platform for the study of epigenetics-related gene silencing. Technique for removal of marker gene production and release of GM crops, or for that matter any genetically modified organism, is always under severe scrutiny both due to scientific, biosafety, and ethical reasons. It has to pass many tests and trials before actually being released in the environment or consumer market as environment and health are the prime concerns for any nation. The apex body in India that evaluates environmental and biosafety activities related to large-scale use of microorganisms and recombinants in research and industry is the Genetic Engineering Appraisal Committee, which functions as a statutory body in the Ministry of Environment, Forest and Climate Change (MOF and CC). When dealing with GM plants, any kind of potential risk that the foreign gene may pose to the environment and health of people is assessed. Anything foreign is not easily acceptable, so is the case with the introduced transgene. Selectable marker genes usually conferring antibiotic or herbicide resistance regulated by prokaryotic or eukaryotic promoters also get introduced into plant genomes while performing transformation. They are necessary to prove the efficacy of the protocol and also distinguish between the transformed and non-transformed cells. But once a transgenic is produced, the marker gene does not have any practical utility. Its occurrence in GM crops, along with the introduced transgene, is a matter of great concern speculating the environmental problems it can have after cultivation and medical implications on the consumption of these GM crops. Biosafety concerns force the need for evaluation of potential impacts of marker genes on human health. Biosafety concerns force the need for evaluation of potential impacts of marker genes on human health and environment. It is thought that although rare, there can be chances of horizontal gene transfer with microorganisms of the gut flora on consumption by humans or animals. Even gene escape by pollen or seed dispersal may occur to the wild and weed relatives, making them more resistant and resulting in gene pollution. Nevertheless, to date, scientific evidence of deleterious effects of the marker gene is lacking. Still, several governing committees state marker gene removal to be a good laboratory practice and recommend that. Consequently, after every transformation event, the need to validate that the marker genes are indeed harmless arises. Reporter genes like GFP have been used as biosensors and are not found to be toxic. So, they can also be used while creating transgenics. Another approach favorable in the long run is to altogether remove the marker genes after the production of GM crops so that the question does not arise at all. Marker-free transgenic plants can be efficiently generated by several new chromosomal location followed by fertilization and segregation of boy and marker. It is successful but has low efficacy and inconsuming. It is not a preferred strategy as excision is not precise and repeated insertions and excisions can cause mutations. Relocation to a new chromosomal position in the genome may also alter the level of transgene expression. Intrachromosomal homologous recombination (ICR) to remove selectable marker gene. The natural nuclear recombination system of DNA repair machinery provides a valuable alternative for a comparatively recently devised novel approach to excise the marker gene. It is based on marker deletion caused by intrachromosomal homologous recombination (ICR) between two homologous sequences. Two direct repeats flank both sides of marker gene to be removed and ICR occurs between them. Homologous recombination repairs the double-stranded breaks. It is a simple technique, removes markers after a single round of transformation and does not involve sexual reproduction, so apt for vegetatively propagated plants. The first time it was elucidated in green alga known as Reinhardti. Thereafter, employed by Peter Meyer's group for transgenic tobacco production in which two selectable marker genes, NPTI marker and TMS2 gene were flanked by two ATTP, attachment P regions of 352 BP of lambda phage and a binary vector PAD ICR. Usually, this ATTP site is used by phage to integrate into the E. coli genome at ATTP region catalyzed by two proteins, the phage encoded integrase int and the bacterial integration host factor IHF. However, this method has found applicability only in some host genotypes and the mechanism is not that clear. Non-target genes may also get deleted as recombination cannot be controlled. Site-specific recombinase-mediated excision of marker. It is an enzyme-based strategy and method of choice for the elimination of marker. Methods like co-transformation, transposition, homologous recombination, and site-specific recombination each will be discussed below. Co-transformation and segregation in future lines. It is one of the simplest strategies for generation of marker-free plants. The GOY and marker gene are integrated into different unlinked locations in the host genome. In subsequent generations, it gets segregated yielding progenies that carry transgene and eliminate the marker. This is made possible in three ways. Two different agrobacterium strains carry a transformation vector with either GOY or marker. A bacterial strain harboring two vectors each with either GOY or marker. A bacterial gene with a plasmid having multiple tDNA regions. Agrobacterium-mediated co-transformation is more advantageous than that mediated by the gene gun, as in the former method, co-transformation genes integrate into separate loci in the host genome. But as the method relies on sexual mode of reproduction, it cannot be used in plants that perpetuate by vegetative means. After segregation, selection of progeny carrying only the transgene is also a laborious task. For e.g., this method has been employed for developing transgenic plants of Oryza sativa, Brassica navis, and Nicotiana tabacum. Transposon-mediated repositioning of genes marker deletion can also take place via the jumping genes or transposable elements which have the ability to jump to different positions on the plant genome. It is analogous to site-specific recombination but uses transposons to dissociate GOY and marker gene in place of recombinase and recognition sites. It can proceed in two ways. 1. The GOY and marker gene is placed within transposon that is lost after transposition. E.g., IPT gene was inserted into the transposable element AC35 for generating tobacco and aspen transgenics. 2. Has mobile transgene that upon transposase activation gets relocated to a region of APP flank both sides of the marker gene. Core region provides directionality to the site. Recombination of DNA between the two recognition sites, i.e., LOXP is catalyzed by pre-recombinase and does not require any other factor. Depending on the position and relative orientation of the two sites, linear or circular enzyme type recombination can lead to site-specific excision. Figure 12.51a integration, figure 12.51b inversion, figure 12.51c or translocation, figure 12.51d. In this case, when orientation is in the same direction, reaction between 3 and 2 LOXP sequences leads to the excision of marker gene. If orientation would be in opposite direction, it results into inversion of internal sequence. Retransformation or crossing introduces the pre-recombinase gene into transgenic plants and excises the insert. It is precise and conservative. The nucleotides of the recombinant site are neither lost nor altered but applicable to sexually reproducing plant species. Gene sequences. There are three methods available for site-specific recombinase-mediated excisions: pre-LOXP system of bacteriophage P1, FLPFRT system from 2 microliters plasmid of Saccharomyces cerevisiae yeast, and RRS system from PSR1 plasmid of Zygosaccharomyces reishi. A common feature in all of these is that two recombination sites oriented in the same direction direct repeats flank of gene. The direct repeats have recognition site for site-specific recombinase. In these methods, LOXP, FRT, and RS are the recombination sites while CRE, FLP, and R are the recombinases. Microbial recombinases can cleave DNA at definite sites and ligate it to cut DNA as a second target sequence. Out of the available methods, CRE-LOXP system is the most widely employed and for the first time used in case of tobacco. The CRE gene has a product of 38 kDa. The asymmetric LOXP recombination sites of 34 BP comprising of two inverted repeat regions of 13 BP and a core. Figure 12.51 site-specific recombinase-mediated excision of marker. Allowing for the selection of transformants lacking marker gene expression at maturity. Conclusion. The gene transfer technology has been a revolutionary step and many methods have been devised for attaining success in the integration of transgene and stable transformants. However, the feasibility of these techniques needs to be optimized for more genotypes and target tissues, and a universal technique applicable to a wider group of plants is always welcome. At the experimental stage, role of selectable marker genes cannot be ignored, especially in case of recalcitrant plant species or wherever transformation efficiency achieved is low. But their usage should be discouraged or restricted owing to potential risks and ethical issues. Other safer marker genes should be given priority. The debate on acceptance of transgenics by the non-scientific community still persists in continuous efforts to meet the demands of the burgeoning population or the need of the hour. The recently discovered technique of genome editing is also a brilliant and imminent alternative for the future. Development of transgenic plant over ages, natural selection and evolution have resulted in the development of plant species with improved traits. Plant breeders have also carefully exploited their knowledge of selecting the right combination of alleles to develop plants with better traits. However, the results obtained were limited. So, since 1970s, scientists have utilized recombinant DNA technology to manipulate the plant genome and develop transgenic plants. A transgenic plant is defined as a plant carrying a transgene that confers a specific desired trait. A transgene is a DNA fragment that has been artificially integrated into the genome of an organism. The development of a transgenic plant comprises of a few basic steps: identification of a suitable transgene expected to modulate an agronomical trait, the introduction of the transgene into the plant, incorporation of the transgene in the plant genome, and its. The CRE-LOXP system, the asymmetric LOXP recombination sites of 34 BP blank codes of the marker gene. Recombination of DNA between the two recognition sites, i.e., LOXP is catalyzed by CRE recombinase. Depending on the position and relative orientation of the two sites, linear or circular, enzyme type recombination can lead to site-specific excision, integration, inversion, or translocation. Modulation of marker gene expression besides excision of selectable marker. Other approaches to deal with the public euphoria can be either not using marker gene at all or developing environment-friendly markers and transplasonic plants or modulating spatial and temporal marker gene expression. If the desired transgene is transferred into an individual zygote directly by microinjection, marker gene may not be required. The transgenic plants obtained after chloroplast transformation, wherein the foreign gene is transferred into the plastid genome and not nuclear genome, form the transplasonic plants. Due to non-involvement of nuclear genome and maternal inheritance of the plastid, they do not pose any risk for the pollen-mediated transgene escape. Also, antibiotic resistance genes get amplified upon gene transfer into chloroplast genome. But the vegetatively propagated plants and those exhibiting biparental inheritance are not amenable to this technology. The marker gene expression can also be modulated and prevented by the antisense technology. Upon introduction of a marker gene cloned in reverse direction, antisense RNA complementary to mRNA transcribed of the original marker gene is produced. They hybridize forming RNA duplex, thus rendering mRNA inactive and can get cleaved by the catalytic RNA like ribozyme. Thus, suppression of marker gene expression can be directed by ribozyme. Also, transgenic plants having selectable marker genes can further be transformed with genes encoding antibodies against the selectable marker gene product. Marker gene expression can also be regulated by tagging tissue or developmental stage specific promoter to the marker gene tropical regions as the agroclimatic conditions favor the perpetuation of both the viruses and their vectors. So, while developing biotic stress resistant plants, a considerable focus was given to choosing appropriate transgenes which will confer resistance against viruses. Multiple strategies have been developed to produce virus resistant plants. The central idea of all these strategies is to identify a viral protein or a set of viral proteins whose variation in spatiotemporal expression will cause hindrance to the infection mechanism of the virus and thus prevent disease establishment in host plants. Based on the type of protein being exploited as a transgene, the various types of resistance mechanisms developed in plants are classified as below. Coke protein-mediated cross-protection. Paul Eagle et al. 1986 first demonstrated that when the coke protein of tobacco mosaic virus (TMV) is used as a transgene in the transgenic construct used to develop transgenic tobacco plants, the regenerated plantlets exhibited resistance to the virus.